Good morning, everyone. Thanks a lot for coming to the first CIC lecture of 2021. I wish you all a very happy new year and all the best success for 2021. It's a particular treat for me this year for this lecture because we get to welcome back one of our lab alum from way back when. Colleen did her undergraduate degree in neuroscience at McGill and spent a couple of summers and better part of a year in my lab working on the impact of high-fat diet on in Alzheimer's disease model, and published a very nice paper on that. And then she went on to become a Gates scholar, where she's been studying neuroscience, through which she's been studying neuroscience at Cambridge, and she's in her fourth year now, so starting to kind of wrap up the whole story. And she's been kind enough to share her findings and her thesis work with us, her thesis work to date with us in this lecture today. So Colleen, it's really nice to welcome you back, even if you can't do it in person. And looking forward to hearing what you've been up to for the last 3.5 years. So take it away.
Great. Thank you so much for the introduction and also for the invitation to give this talk. It is really a privilege to be giving this talk because, as Malar mentioned, I had some of my very first research experiences at the CIC, where I worked with Malar in the COBRA lab. And this is really, I think, where I began learning what it means to be a neuroscientist, and more importantly, where I worked with some really wonderful people and had a lot of fun doing science. So I consider myself lucky that this was my starting point, and it's nice to be returning back to give this lecture. So a bit about me, as Malar mentioned, I did my undergraduate degree at McGill in neuroscience. And this fueled my rather broad interests at the time and how changes to the brain's functional organization or structural organization affect mental health. I did my thesis project with Malar, looking at different lifestyle factors for Alzheimer's disease, particularly obesity in a mouse model of Alzheimer's disease. And in this, I learned a lot about the tools and some of the methods that are available to study the brain's organization in health and in illness. I then went across the pond to explore some of these questions about the brain's organization in more depth. And I'm now in my 4th and final year of my PhD at the University of Cambridge in the Department of Psychiatry. Rather broadly, my research explores how the brain supports the experience of hallucinations, mostly in people who have a diagnosis of schizophrenia. And to study this, I use structural and functional brain imaging, use some cognitive tasks, and also now I'm moving towards some qualitative research on the lived experiences of hallucinations in different disorders. So this was one of the starting points for my PhD research. In 2015, my colleague and mentor, Jane Garrison, published this paper as the outcome of some of her own PhD research on how parasingulate sulcus morphology is associated with hallucinations in the human brain. She approached this research from both a psychology and a psychiatry perspective. She was interested in this construct known as reality monitoring, which is essentially a decision-making process that's involved in distinguishing between internally generated information like visual imagery to information generated in the external world, so a sound that has a direct cause from an object. It's essentially the process that answers the question, how do you know what you're experiencing in terms of your senses is actually real and actually happening? And there's evidence that people with hallucinations show reduced capacity in this ability to monitor reality. And previous work in her group had also shown this association between reality monitoring and parasingulate sulcus morphology. So the parasingulate sulcus is this sulcus or brain fold that lies dorsal to the cingulate sulcus in the medial prefrontal cortex, if it is present, though it shows a lot of inter-individual variability between people. I think this was an interesting finding because it showcases perhaps a fundamental principle that the way we perceive and experience our surroundings in adult life can, in part or to some degree, be influenced by how the brain develops in utero. I think it shows that there is a structural biomarker that was developed in utero for a complex aspect of psychopathology in adulthood. And so one of the questions that my research has been trying to answer is, so what? What does this seemingly random biomarker in the prefrontal cortex have to do with hallucinations and schizophrenia? And just to show a few more findings from the original research study that she did. This was in the Australian Schizophrenia Research Bank, around the scale of 100. So there were 79 patients with hallucinations and 34 without. And she found that this reduced length of the parasingulate sulcus occurred in both hemispheres, but only in the left for this hallucination to no hallucination comparison. And she also found association with other aspects of brain morphology. So patients who experienced hallucinations had increased gray matter and reduced stratification in its prefrontal region. So what? How can properties of sulci influence cognition? How do sulci develop in the brain? And what does this all really mean? Well, we know that sulci emerge in the 2nd to 3rd trimester of pregnancy, and they emerge in a very specific temporal order. So these primary sulci develop first, and these are under stronger genetic control, and they're also more stable across individuals. So for instance, the cingulate sulcus is a primary sulci, and this should be present in everyone's brains unless there's some malformation, and it should also be in relatively the same place in everyone's brains. However, these secondary and tertiary sulci develop later in utero. They have weaker genetic influence, but stronger environmental or in-womb factors that influence how they are formed. And they also show much higher inter-individual and also interhemispheric variability. So this parasingulate sulcus, for instance, it can be present or it can be absent in different individuals, and this presence or absence can also occur in different hemispheres. So you might have a PCS in your left hemisphere, but you might not have this specific sulcus in your right hemisphere. So these are very variable sulci. We also know that sulcal patterns are stable across the first few decades of life. So for instance, in this study by Arno Cascia in 2016, he showed that the sulcal pattern of whether or not you have a parasingulate sulcus, which is also called a double cingulate because it's the sulcus above the cingulate sulcus. This pattern is stable from late childhood, so if you're around 11 years of age, into early adulthood around mid-20s. We know that certain features of the sulci can change. So for instance, it's common to have a sulcal widening with age, but what is common across at least this period of life is that the pattern of whether or not you have the sulcus and sort of the relative shape is pretty constant. And this is in contrast to other structural features of the brain, like cortical thickness or gray matter volume, which we know are very plastic to aging, to learning, and to experience. We also know that inter-individual variability in sulcal patterns influences local cytoarchitecture, it influences functional organization, and it influences cognitive processes in health and disease. And the influence of sulcal patterns on cognition can be anywhere from very striking to very subtle. So for instance, this so-called power button symbol is a specific sulcal configuration that's associated with type 2 focal cortical dysplasia, which is a brain malformation that results in epilepsy. And this pattern is present in something like 1 out of 40 healthy controls, but it's present in about 2/3 of patients who have type 2 focal cortical dysplasia. So it's a very strong indicator of specific behavior and cognition. On the more subtle side, sulcal organization influences the somatosensory representation in the motor cortex. So the specific sulcal patterns influence where in the brain you have your lip representation or your hand representation. And also other studies that have looked at the parasingulate sulcus have shown that whether it is present or absent influences cognitive control processes, specifically inhibitory control, both in children and in first episode psychosis. Sulcal patterns also influence the brain's functional organization. So here was a very relevant study done in 2019 by Lopez Person. And here the authors looked at how the sulcal organization of the ventromedial prefrontal cortex influenced the activity of the default mode network. And as you can see here, they found that groups of subjects with and without this specific sulci, in this case the RRSI, had very different patterns of default mode network connectivity. And this might suggest that this is a general principle where sulcal organization influences some of these large-scale networks like the default mode network or the salience network or auditory and visual networks. And this is particularly important to my research because hallucinations are associated with both broad and specific changes in functional connectivity. And schizophrenia, more in general, can be thought to be of a syndrome of disconnectivity of different brain circuits. So some past research on hallucinations have shown that they're associated with alterations, both widespread and focal, in different functional networks. Particularly, they've been associated with alterations in the language networks. And I should add that this is more specific to hallucinations in schizophrenia, because these are often auditory hallucinations, which can manifest as voices saying usually pretty unpleasant and distressing things to the person. But it's important to recognize that hallucinations can also occur in other modalities, such as visual or such as touch or smell. And they also occur in many other disorders besides schizophrenia. Besides language networks, ones particularly involving the anterior cingulate or self-referential networks have been involved in hallucinations. Here on the left is a meta-analysis of different studies of this construct reality monitoring, showing that the peak fMRI activation associated with tasks that assess this function are mostly within the anterior medial prefrontal cortex. A rather broad and perhaps overly simplified hypothesis about hallucinations is that there is overactivity within these sensory regions, such as the auditory cortex, which these medial prefrontal regions, which are more associated with self and recognizing self from other or self from environment, have a reduced activity. And this is some earlier work that I had done just to emphasize this point that there are different routes to hallucinations. They have different causes and likely many different mechanisms. So this was a meta-analysis on a transdiagnostic perspective of brain changes with hallucinations. And I had found that people with a psychiatric hallucination, so this was mainly schizophrenia or bipolar disorder, had more of this frontotemporal pattern of gray matter associated with hallucinations, whereas people with a neurodegenerative condition like Parkinson's or Alzheimer's had more changes in the occipital cortex. But the research I'll be presenting and talking about now is more focusing on these psychiatric hallucinations, primarily hallucinations and schizophrenia. So the objectives of the research, the first one was replication. Is the reduced parasingulate sulcus length a replicable biomarker for hallucinations and schizophrenia? I next wanted to look at different methods to assess sulcal morphology. I also wanted to look at the superior temporal sulcus, which is in the auditory cortex and has also been associated with hallucinations. And then I was interested in this question of how these specific sulcal changes how does that associate with the whole brain, which we really believe to be involved in hallucinations of brain networks, not just sort of these focal structural changes. And then I hypothesized that, like previous research, that I would find a reduction in the parasingulate sulcus length, that the superior temporal sulcus might also show morphological differences, and also that the variance between different brain regions associated with networks involved in hallucinations might show alterations as well. So these are the two data sets that I've been working with to look at these objectives and hypotheses. They've both been repurposed from previous studies. So the first study was part of a multi-center trial in the UK to explore the benefits of minocycline to treat negative symptoms in schizophrenia. It was a longitudinal study, but I've only used these scans from the baseline MRI before patients were treated with minocycline, which unfortunately was not effective to treat negative symptoms, but that's another story. Oops. I also only used the first 3 largest centers because the other ones had different demographics slightly. The second study was from collaborators in Shanghai, and they were interested in the striatal dopaminergic system in schizophrenia and how people respond to different monetary and affective incentives. But likewise to the first one, I just used these first scans and their symptom scores on different psychiatric ratings. So for both data sets, I stratified them into patients who did and did not experience hallucinations based on their PANSS P3 score item for hallucinations. And I should add a caveat now that this is a big limitation, I think, to the research, because the PANSS only measures symptoms within the past week, so the past week before, since they were scanned. And it's a scale from 1 to 7 for the severity of those hallucinations. It's also not modality specific. It's sort of just a general question of, do you see or hear things that aren't there? So for this research, there is the assumption that these were likely auditory hallucinations, because hallucinations are experienced by about 60 to 80% of patients with schizophrenia in the auditory modality. So looking at the 2 data sets, fortunately, they were relatively similar in terms of the demographics, in terms of the male-female ratios. The mean age for both was around 25, so these were young patients, either first episode or in the early stages of schizophrenia. The hallucination had a PANSS score of around 4 from the scale of 1 to 7, whereas those without hallucinations, it was from 1. They had no significant differences in their medication. There were significant differences in their delusion scores, where those with hallucinations had more severe delusions, but that is to be expected because there's a lot of symptom overlap between hallucinations and delusions.
Colleen, we have a quick question. Did you look at anything related to, this is from Ranjini, do you have anything related to age of onset of the symptoms that you've collected here? Or
no, unfortunately, I think that was one of the variables that was very poorly characterized.
Okay.
Yeah.
Thanks.
And briefly for the scanning parameters, they were all 3 Tesla scans. The multi-center study in the United Kingdom had a protocol to harmonize their different scanning parameters. The Shanghai one was a separate study, so it was not part of that, but it was also 3 Tesla and had relatively similar scanning parameters. So before I get into some of the findings from my research, I just want to illustrate some of the challenges of measuring these sulci in the brain. So you can see here that the pattern variability poses a real challenge to both identification, classification, and measurement. So sulcal patterns can vary across individuals and between hemispheres, both in terms of the shape or the position of the sulci. They can either be a continuous sulci or they can be broken up into different segments. And they can even be present or absent. So one sulci doesn't have to be present in all individuals. You can see here, this is just one sulcal pattern, or 3 different sulcal patterns in the same brain region, and they can vary substantially between individuals. So the measurement techniques for sulci, the gold standard for most of these tertiary or secondary sulci, is still manual segmentation, which, as many of you know, is very laborious, time-consuming, and can be a bit of a subjective process. There are some automated softwares to recognize and identify sulci, which can perform quite well for the primary sulci, such as the cingulate sulcus, which is more stable across individuals. But they're not so good for identifying these more variable sulci. So here's an example of the superior temporal sulcus in the auditory cortex. And you can see here, the sulcus in one individual is very continuous and whole. The other 2 in other individuals are broken and sort of twisted in different ways. And these representations were created with a software I've been using called BrainVisa. And this is a sulci that creates these cortical fold graphs. It's very good at identifying different sulci in the brain based on the gray matter, white matter contrast in the MRI scan, but it's not so good at labeling these variable sulci like the parasingulate sulcus. So for these 2 data sets, I've used both a manual segmentation and this BrainVisa software to measure the sulci that I'm interested in, namely the parasingulate and superior temporal sulcus. So BrainVisa first does bias correction on T1 images. It does brain extraction and splits the brain into the left and right hemisphere and cerebellum, and segments these into the different components of gray matter, white matter, and CSF. It then creates this, I guess, skeletal structure of the different sulci in the brain. And this, I guess you can think of it as a graph of the different sulci, recognizes the different folds and also their mutual relations. It then also tries to identify different sulci in the brain, but it is not so good at labeling the sulci that I was interested in, namely the parasingulate sulcus. But the outcome of this is that you automatically get different features of the sulci, such as the length and depth, and also the surface area and other features as well. And this is interesting because this is not really possible to get for manual segmentation. You can only really get length of the sulcus for manual segmentation. So after BrainVisa creates these graphs of the sulci, I've created this semi-automated method where I then label which of the sulci that is identified is the parasingulate sulcus.
Colleen, I just have a quick question. To what extent do you have to do kind of some retraining of the software package to get this done? Or was it kind of really just taking the outputs and then repurposing what's going on in that, based on the labels of that specific region? Do you, your semi-automated method, does it kind of just involve kind of looking at a region of interest and then saying everything within this should be at some level labeled the sulcus?
I would say it's pretty straightforward usually. The pre-processing steps to create these sulci graphs, the software is pretty good at that. It is pretty good at seeing what is or isn't a sulcus in the brain. But for the labeling section, it's kind of more user interface, explore the graph and find the region that you're looking at, and whether or not you think it's the parasingulate sulcus.
Okay.
That makes sense.
Yeah, it does make sense. And then when you look at kind of the morphology of the sulcus, like in terms of, branching and, the shape obviously comes probably nicely out of the BrainVisa outputs, but anything more in terms of the continuity of the sulcus, is that something that you have to do yourself, or does that kind of come out as an automatic output as well?
It gives different metrics, but in terms of the shape and the position to the rest of the brain, that is more up to you. So you can get this as like either a volumetric or a surface-based shape, and then you can do other things with that. But yeah, that would be beyond the software.
Okay, great, thanks.
And yeah, so just following that, this is looking at the manual and the semi-automated method. And they have very high iterative reliabilities, about 0.93 or 94. But this is a bit to be expected because the user involvement is the same for both of these. So both in the brain visa and in the manual, you're selecting which sulci you think is the parasingulate sulcus. And I've been doing this work based on different protocols that have been developed earlier to identify these sulci. So the first result that came from some of this work is that the length of the parasingulate sulcus was reduced in the left hemisphere for both of these data sets that I've worked with in patients who experience hallucinations. And this was, to be honest, pretty surprising considering the lack of reproducibility in brain morphology in a lot of different psychiatric illnesses, particularly one as heterogeneous as schizophrenia. What I thought was interesting digging into these results further was that this reduction in the length actually reduces the asymmetry index between the sulci. So in the general population, there's a really well documented fact that the parasingulate sulcus shows this leftward asymmetry, where it is either longer or more prominent in the left hemisphere than that of the right. But as you can see here in this graph on the right, this asymmetry is just completely reduced in patients who experience hallucinations, which I think is a new finding adding to this literature. And we know how important asymmetries are in the human brain in relation to language and other cognitive processes.
Sorry, Colleen, one more question.
Yeah.
Why are there no, perhaps you mentioned this and I missed it, and I apologize if that's the case. Why are there no controls in the UK sample on the length estimate?
Yes, that was just the data set that I was using. It was from this multi-center trial looking at the effects of a specific drug on negative symptoms of psychosis. And so they didn't recruit healthy controls for the purpose of their study, which is fortunate for me.
I have another question. Does no hallucinations indicate absence of hallucinations for a particular period of time, or are the patients in remission?
That's a good question. It indicates the first, so no hallucinations sort of within the past while. We think it correlates well with never having experienced hallucinations because the patients in these studies were either first episode or very earlier in their psychosis. So if they're not having hallucinations now, it's likely that they didn't before they were psychotic. But it is a bit of an assumption, and it could be that this is more a marker of treatment resistance.
And then maybe it's a better question to ask at the end, but what do you make of the specificity of these findings? You know, like to what extent do you think this is a marker of hallucinations rather than overall severity, for example? Can you rule that out? Or even other, or even other positive symptoms like delusions or something like that.
Yeah. I don't think it can be ruled out completely. I did control for overall psychopathology, so overall positive symptoms, and it wasn't significant for that. So it seems to be specific for hallucinations, but I think what would be needed to really confirm that is probably a better symptom score or a better marker of hallucinations. So maybe like lifetime history, something a bit more strength than just the PANS, which is just a brief symptom history.
Thanks.
And then the next finding, I looked at the superior temporal sulcus. And I didn't find any changes for the length, but I did find that patients with hallucinations compared to healthy controls had a reduction in the depth in the right hemisphere. And this is also related to asymmetry because the superior temporal sulcus in the general population normally shows this rightward depth asymmetry, where it's deeper in the right hemisphere than that of the left. And so this was slightly more reduced in patients with hallucinations, though not to the same degree as the parasingulate sulcus.
So I have a question from Gabe. The length measure, is it Euclidean length or geodesic length along the curve?
That would be geodesic, yes.
Thanks.
Yes, the entire length of the sulcus, with including all its curves. So I then wanted to look more at the shape of these different sulci to see what is causing these different differences. So for instance, is the parasingulate sulcus, is it shorter because it's more broken up into different segments, which is quite often something that I see. So to do this, I transformed the different sulci into standard MNI space and overlay the group sulci to create these sort of group-wise average maps of the sulci to see what the average shape of the different sulci look like. And you can see here that there are clear differences in the shape of the structures, and you can see kind of a kink, I would call it, especially for the superior temporal sulcus. And for the PCS, it looks like it's more broken and more direct in people who experience hallucinations. And this showed a very striking parallel to previous research that had been done, which had shown that the location of the sulcal junction between the right superior temporal sulcus and its anterior branch was associated with inner versus outer space hallucinations, which refers to whether hallucinations are heard as inside the head, so more like thoughts, or whether they're heard as outside the head, so more like voices of someone speaking towards you. So it really seems to be beyond just length and depth. It seems to be the shape and the position of the sulci that's also important. I next want to explore how these specific sulcal anomalies fit into whole brain development and whole brain connectivity, because we know that these brain networks are involved in hallucinations. I was advised, based on work by previous students in our department, to look at structural covariance. And structural covariance is the statistical association between pairs of brain regions and their anatomical properties across the population. So for instance, if you take one of these brain regions X and this brain region Y, and for instance, if you calculate the gyrification within both of these brain regions, you correlate those for all participants in that study or all subjects in that sort of group of patients versus controls, and you end up in this. You end up with this matrix with the number of brain regions by the number of brain regions. And each cell in this brain region would correspond the group-wise average correlation of that metric between those two brain regions. And there's mounting evidence suggests that these structural covariance networks might reflect developmental coordination between different brain regions, such that areas with highly correlated anatomical properties like gyrification might result from similarities in their ways that they mature.
So, Colleen, I got another question here. If we go back a couple, just the first set of results, Laurie has a question where he's asking, could local gyrification index be another method to look at local changes like this?
Definitely. And actually, I was just about to say that that is
conveniently, I will let you go on.
Yes, that is actually the metric that I used to look at structural covariance, because structural covariance is typically done by using one of the different brain parcellations. So for my work, I used the Human Connectome Project Atlas developed by Matthew Glaser in 2016, which has 180 brain regions per hemisphere. And it's not really possible to parselate sulci in the same way that it is other structural features of the brain. So instead, I've used the local gyrification index, which reflects the amount of cortex in a sulcal fold compared to the outer surface of the hemisphere. And this is also thought to be an imaging marker of brain development. And it's more sensitive than other curvature-based markers of development. For instance, a study in 2016 showed that it's more sensitive than other markers to differentiate preterm infants with brain injury from those without. And we know that preterm birth is often associated with changes in brain folding. So using the local gyrification index, which I hope is a proxy for sulcal folding or brain folding, I created these group-wise structural covariance matrices for each of these three groups of patients with hallucinations, those without, and healthy controls. I then averaged the different brain structures that correspond to different large-scale brain networks to get the mean local gyrification index for each of these different large-scale networks. I was specifically interested in the salience network and the auditory network based on previous research that has shown their involvement in hallucinations. Because these covariance matrices represent group-level data, I used a permutation approach, which essentially randomly reshuffles these participants to different matrices of the same size as the original groups. And it does this, say, 10,000 times. And it plots the values across this distribution. And then you see whether the observed value, where that relates on the distribution, if it's in the 5% mark. And this was also corrected for FDR. And I found that the mean local gyrification correlation for the salience and auditory network, and also between these two networks, was increased in the group who experienced hallucinations compared to those who did not, and also to healthy controls. And I think this suggests that something about the way that the sulci form in utero has some sort of effect on developing brain networks. So this is not necessarily a new finding. I mean, we know that schizophrenia has long been considered a neurodevelopmental disorder since the proposal in the 1980s by Daniel Weinberger. And there's also been an increasing number of studies, especially in the last few years, about how cortical folding alterations are related to schizophrenia. What I think is new, however, is that it's showing that these specific sulci, which are also related to cognitive processes like reality monitoring and audition, are related to hallucinations, and also that these specific folding patterns relate to large-scale networks, which we also know are involved in hallucinations. So I hope I've elaborated some detail on how this finding of a structural biomarker relates to hallucinations, but the question still remains of how these cortical folding patterns lead to these complex experiences as adulthood, which I think can really only be answered with longitudinal studies characterizing brain folding in utero to experiences in adulthood, which hopefully will be possible in coming years with improved developments in fetal MRI. Like many research findings, I think that this raises more questions than it answers. Both about the etiology of hallucinations and also the real-time mechanism for how they occur. Based on the data that I've had, I've done more research on this latter one about the mechanism. So for both of these two data sets that I've studied, they also had available resting state data, which is something that I'm looking into now, whether these sulcal findings have any relation to the functional activity in the brain. I don't know if I have time to really get into these findings, but just a preliminary look at them. I've pre-processed the functional data with these pre-processing steps here. And I've also been using the HCP atlas of 180 brain regions per hemisphere to parselate this. Then extracted the regional time series for each of the 360 regions, and as well as 16 subcortical regions. And I estimated the functional connectivity by computing the Pearson's correlation coefficient between the regional time series. I also did various quality control metrics, including visual QC, and also calculated mean frame-wise displacement and DVARs to look at motion.
Colleen, before you get onto these results, we have a question from Lena Palinapon, and he's asking, when the PCS is interrupted, how did you calculate the length? Is the interruption more important than the length per se for hallucinations?
So when the PCS is interrupted, we measure just the fragments. So one fragment here and one fragment here, and then the length is just considered as the total length of the fragments. So you don't include the gap between them, you just include the total length. I haven't done anything to explore whether the fragmentation in specific seems to be important. I mean, from the images of the Sulcal maps, it does seem that at least the position of where these fragments are seems to be important. So I would think that whether it is fragmented or not would be, but I don't know anything about specific number of fragments or anything like that.
Thanks.
So just briefly for the functional connectivity, there haven't been any striking results that I've looked at so far. So this is looking at both the mean resting state functional connectivity between the sulci that I was looking at, so between the left PCS and the right SDS. And this is also at 2 different functional connectivity bands for both the Shanghai dataset and the UK dataset. The only difference that appear to be are between healthy controls and the patient groups. And on the right, I was looking at the shared interregional correlations between these 2 sulci. So if you imagine that the parasingulate sulcus is connected in different ways to all the other brain regions, and the STS is also connected to all of the brain regions, this metric is looking at what percentage of brain regions are common to both of them as you increase the threshold of functional connectivity. And as I mentioned, it really only appears to be between healthy controls and the patient groups. So perhaps this is less discriminative a marker than other measures of brain function or structure. And just briefly on the more etiological side of things, I haven't done any research on this because I don't have any developmental data, but I think it's very interesting to consider because I think the findings obviously have implication for brain development. We know that there are many different neurodevelopmental processes that occur in the 3rd and 2nd trimester of pregnancy, such as neuronal migration, synaptogenesis, pruning. And we know that these neuronal events, which are partly under genetic control, are also influenced by in-womb environment, such as maternal stress or alcohol and drug intake, or hypoxia or infection. And I think that these in utero factors could influence the healthy fetal brain with lasting implications on human development in cognition and functional activity. There's some evidence of the question of how this cortical folds results in the psychopathology we see in adulthood. So one hypothesis is that the cortical folding patterns might influence how different white matter tracts develop. So they could influence both structural and functional connectivity. And this was a recent study published by Falkenberg in Psychiatric Research that shows that patients with frequent hallucinations, that's in the figure A on the left, had longer reconstructed tracts in the arcuate fasciculus, which connects Wernicke and Broca's area, compared to patients without hallucinations and to healthy controls. So there is some evidence that these cortical folding patterns might later interact or influence structural and functional connectivity. So these are some of the more questions that I think my research has raised and that I'm interested in. Are sulcal anomalies modality specific, or is it a general mechanism for hallucinations? So do these just occur for auditory hallucinations in schizophrenia, or do they occur for other types of hallucinations in different conditions? Do they occur for other disorders, particularly ones that have neurodevelopmental influences like borderline personality disorder? Do they interact with aging? For instance, we know that visual hallucinations commonly happen in Parkinson's, especially as the disease progresses. Do these neurodevelopmental outcomes, how do these interact with and mediate life experiences and other risk factors for hallucinations like trauma? Does this network of sulcal anomalies have any predictive value? Does it predict whether you will convert for high-risk individuals to having psychosis? Or does it predict anything about the trajectory of your symptoms? Also, from a neuroimaging perspective, we know that these variations in sulci are important for interpreting functional imaging outcomes. And finally, what are the mechanisms underlying cortical folding, and how might this constrain or blueprint functional connectivity? And just my last slide. Regardless of what the different mechanisms are that influence cortical folding, I think it's important to recognize that long-term outcomes of altered cortical folding on cognition and psychopathology. So these brain findings that I've identified in the different sulci are just one of the many risk factors for experiencing hallucinations in schizophrenia. But I think it's a very interesting case example of how the outcome of these cortical folding processes, things that happen in utero, can influence how we experience the world much later in life. And this was just one example of a recent study that showed differences between preterm birth and how that later affects psychopathology. And I think that to answer these questions, we'll really need some longitudinal studies characterizing how folding patterns in the brain might scaffold functional connectivity and life experiences. And so finally, I just wanted to thank everyone who's mentored and supervised me over the course of my PhD, particularly my supervisors at Cambridge, John Suckling and Dr. Graham Murray, as well as my mentor, Jane Garrison, also Professor John Simons, and a few collaborators I've worked with, Dr. Liliana Galindo, Dr. Raymond Chan, and Professor Arno Cascia. I'd just like to add that most of the research I had presented on these Sulcal findings have been presented in this paper that was recently accepted in Translational Psychiatry. So if any of that interests you, please do go ahead and read it. And I'd also like to thank my funding body, Gates Cambridge, and I'd also like to thank the Cobra Lab for supporting my development as a neuroscientist. So thank you so much for listening.
Thanks for sticking this in there, Colleen. I appreciate it. Wonderful talk. I mean, a really nice background and really interesting findings, and I like how you tied it together at the end with kind of future developments and ideas for that. So we have about maybe about 10 minutes for questions. We have two already in the chat. One is from Gabe, and the question is: are the anatomical anomalies, by which I mean, I guess he means the differences in folding and gyrification, a causal or correlative anomaly with underlying causes?
I'm sorry, could you repeat that?
Yeah, I don't know. Gabe, you wanna take this one maybe?
Sure.
I'm not exactly sure that I'm doing it.
So what I'm asking here is you're kind of I kind of get the sense you're implying that the sulcal abnormalities you're finding are causing hallucinations. but I'm asking whether or not you have any evidence of that, or whether there might be something else underlying it, and how might you answer that?
Definitely. No, I think that's very important to recognize that it's definitely not causal, or at least none of my research can indicate that it's causal. I haven't looked at any data from, early brain development to psychopathology in adulthood. What might lead me to suspect that it has a role either in then influencing, how the brain forms and how the brain interacts with and influences and is influenced by life experiences, I think is an interesting question. And we know that these sulcal patterns that I've identified, so the shape of the PCS and where it is in the brain, some research has showed that that is probably stable across the lifespan, or at least until early adulthood. So if that's a marker that's sort of established in utero and it doesn't really change across the lifespan, I think that gives, I guess, some validity that these neurodevelopmental markers at least have some sort of association with psychopathology.
There's another question from Trizana. In the patients who did not have the Parasingulate sulcus, how many experienced hallucinations?
That is a great question, and I don't know the answer. I didn't look at specific cases. So these are essentially averaging the lengths of the parasingulate sulcus for groups with and without hallucinations. That would be interesting, though, to know both what percentage had completely no parasingulate sulcus and whether that has any effect on hallucinations. But I'm afraid I don't know the answer.
I've some follow-up questions as well. Have you looked at or thought about looking at, because you did this kind of connectivity profile with respect to the I guess the functional connectome. Are there any thoughts of adding in data from the structural connectome? So looking at, say, how diffusion data may be different, in the different groupings that you've developed. And because I mean, if one of the hypotheses is that this, is a kind of going back to Gabe's point, this might be the consequence of kind of multiple developmental processes, right? And if kind of axon development and white matter tract development is kind of one of those, you would expect that the connectivity from that PCS region, for example, would be different across your groups. So any thoughts on that?
Yeah, I think essentially exactly what you've been saying. I haven't worked with DTI data myself before, and I don't know of any data sets for that, but I do think also that is the direction that some of this is pointing towards. So yeah, do these cortical folding events, how does that then result in this is really the key question.
Yeah.
And one hypothesis could be that it influences how these different white matter tracts are formed. And there is some evidence in the literature that there are alterations in these white matter tracts. Particularly, people have looked at the arcuate fasciculus, but I haven't worked with it myself.
Someone is with a hand. Gabe, go ahead.
So I just had one suggestion for a kind of a really interesting control group for this kind of work. It's my understanding from the literature that there's never been a case of schizophrenia in a congenitally blind subject. So it'd be interesting to see how the brain anatomy looks in the sulcal anatomy looks in a congenitally blind subject in comparison to, say, normal controls or to schizophrenia subjects.
That would be interesting. Probably very, very rare. Yeah, it's interesting. There's been some research on auditory hallucinations in deaf people. So it's interesting that you bring that up as a control, because I don't really understand how or what that experience would be like. But it's interesting to think about these rare cases.
So, I mean, your group also is. Oh, I have another question that's not mine. So the parasingulate sulcus, this is from Trezanigan. The parasingulate sulcus was thought for the longest time to be human specific, but a recent study published last week. So here you go. Now you're on the hot seat. How good is your capacity to keep up the literature? Has demonstrated its homologue in the chimp. Wow, here we go. You think that chimps hallucinate? And go.
Wow. Yeah, I have read that study actually. I thought it was really interesting too, because it at least showed that this is a higher cognitive process, but I have no idea if chimps hallucinate. I think there's been some studies trying to induce hallucinations in mice, but I know nothing about primates or anything like that. I'm sure there are some videos on YouTube that would indicate whether or not they do.
Yeah, exactly. And yeah, I mean, that's probably a really nice place to stop. So once again, just a fabulous talk. Thanks for sharing your work with us and taking the time to come and speak with us. For those of you who are interested, we are there's a student session after this. So if anyone wants to hang out and chat with Colleen about her work or have any questions to her about life in Cambridge even or anything like that, you know, she's a very excellent resource and very knowledgeable. So if you're interested, please feel free to stay behind. And I will leave so you can speak to her, you know, without any old man supervision. So thanks again, and those who will stay, I'll pass the torch on to someone else so they can host the meeting.
Thanks so much.
Thank you.